Okay. Okay, we can start the class now, right? Up till now, what we have studied in the classes, we have talked about the introduction part of this chapter, and then we have discussed about the anaerobic respiration. Respiration could be broadly divided into aerobic and anaerobic. Anaerobic and fermentation is a part that we have discussed. We have started with the aerobic respiration, and the first step of aerobic respiration is glycolysis. So, we have completed glycolysis and we are almost at the middle of Krebs cycle. From the discussion that we have done in the last class, what we remember, there are certain terms associated with respiration. No, I think you all are now, you know the meaning of complete oxidation and incomplete oxidation, right? Fermentation hole duta fermentation of a difference ki, right? First step of respiration is glycolysis. Glycolysis is oxygen dependent, yes or no? Obviously no, right? Because it is independent of oxygen, because oxygen kuno consume no hoy aru carbon dioxide produce no hoy, right? As far as glycolysis being the first step, what is the product that is synthesized? Whenever we talk about complete oxidation, we know that the respiratory substrate needs to get divided, or it needs to get broken down, and ultimately ki produce koribo lage? Carbon dioxide, ATP and water, right? Out of the three products, in case of glycolysis, which is a 10-step reaction, that key produce hoy, it is only ATP. Again, with ATP production, we have two words, either ATP is produced via ETS or ATP is produced in the substrate level phosphorylation. In the substrate level phosphorylation, we have seen the synthesis of 2 ATP, actually it is 4, but since 2 of them gets consumed in the first and the third reaction of glycolysis, the summation is obviously 2. And next, what is left over is via ETS. Via ETS, glycolysis of Keta produce kore sota, because we know that we see the evolution of 2 NADH2, and 2 NADH2 will ultimately give you 6 ATP. So, the total number of ATP that is produced in glycolysis is 8. Next point is, where does glycolysis occur in a cell? It occurs in the cytoplasm, which is also called as the cytosol, right? So, the first step occurs in the cytoplasm. The next step of aerobic respiration, that is Krebs cycle, it occurs in the matrix of mitochondria. So, we need to have something that links between the first step and the second step. What we are talking about is a link reaction. What happens in glycolysis? Glucose gets divided to form 2 pyruvic acid, but pyruvic acid form kora loge loge, we cannot be saying that respiration process is over. What is the reason that the entire process does not stop at glycolysis? We know that 3 things needs to be synthesized, ATP, carbon dioxide and water. It is only ATP that is synthesized in glycolysis, and so baki kita bostu produce koribo lagile pyruvic acid akko bhangi jabo lagibo. The pyruvic acid needs to get broken down again, right? So, what happens to pyruvic acid? Since the next step is Krebs cycle, pyruvic acid actually should have entered inside the matrix, but we know it cannot enter as such. So, pyruvic acid gets converted to acetyl coenzyme A. Where does this reaction occur? It occurs in the perimitochondrial space. What is perimitochondrial space? The space between the 2 membrane of mitochondria is called as perimitochondrial space. So, this reaction is called as link reaction. Again, if you are talking about a reaction, so obviously this reaction is a part of respiration. A respiration or protective step, protective reaction or ki hobo ATP synthesis hobo. So, in link reaction also we see the formation of ATP. What is the number of ATP per pyruvic acid? It is 3. Eta NADH2 produce hoy. If I say what is the number of ATP produced from pyruvic acid in link reaction, tetihale answer ki hobo? 6, right? Karon duta hoi jabo na, pyruvic acid duta form korisile. Next is acetyl coenzyme A keta form koribo, obviously 2 from 2 pyruvic acid, and acetyl coenzyme A will ultimately enter inside the matrix, right? Once it enters inside the matrix of mitochondria, oxaloacetic acid would be there inside, and it would act as a acceptor molecule, entrant molecule of Krebs cycle or acceptor molecule of Krebs cycle. Entrant molecule ki hoy? Acetyl coenzyme A, যোনটো নেকি পেৰিমাইটোকন্ড্ৰিয়েল স্পেচৰ পৰা কোলে খুমালে ইনসাইড অফ মাইটোকন্ড্ৰিয়াত খুমালে, ৰাইট? আৰু খুমালে এইটো নহব, someone has to take it also, and that is what is oxaloacetic acid. Ultimately, oxaloacetic acid is a 4-carbon compound, acetyl coenzyme A is a 2-carbon compound. Both of them combine together to form a 6-carbon compound, that is a called as citric acid, and so the cycle is also called as citric acid cycle, right. So I think up till that part we have already discussed, right, and we have written certain points about Krebs cycle. Krebs cycle or key monotrakhibo. First, where does it occur? Second, how many rounds of Krebs cycle are necessary for the breakdown of one glucose molecule? 2 rounds of Krebs cycle are necessary per molecule of glucose. Third, what are the products that are synthesized? Obviously ATP to produce koribo. ATP production or logot ako duta word substrate level and via ETS. Substrate level in case of Krebs cycle, we will be drawing the figure today. We have seen that there is the evolution of just 1 GTP per TCA. What is TCA? Tricarboxylic acid cycle, which is also called as Krebs cycle. Krebs cycle kebar hobo laibo dubar. So GTP keita produce koribo duta. Kintu 1 round of keita produce koribo eta. So that is substrate level. Via ETS, if you are talking about ATP kune produce koribo, reducing cofactor bilake produce koribo, and reducing cofactors are NADH2 and FADH2 that are synthesized in the Krebs cycle. You have to know as to what is the number of NADH to produce and what is the number of FADH to produce. Talking about NADH2, it is per cycle ki hobo 3 ta, or FADH2 just 1. Next, NADH2, FADH2 will break down, and ultimately what they will synthesize is ATP. NADH2 eta molecular pra keta ATP produce hoy 3 ta, or FADH2 eta molecular pra keta produce hoy 2 ta, right. Next is, apart from these ATP, ATP uporiu to kiba eta bostu aru synthesized hoy, we have to talk about the role of carbon dioxide evolution. Out of the 3 products, I think you all remember we have said this statement, ATP, water and carbon dioxide 3 ta bostu produce koribo laibo no. Tar bitor synthesis of ATP, credit goes to whom? All the steps of respiration. If you are talking about evolution of carbon dioxide, the credit entirely goes to Krebs cycle, because maximum number of carbon dioxide has evolved during Krebs cycle. And if you are talking about the evolution of water, diba credit to the 3rd step, that is called as terminal oxidation, right. So hekin loke ami korisu. Next, another point that we have discussed is something called as the amphibolic pathway. As a whole, respiration of ki boli kom, catabolic ne anabolic. Obviously catabolic because the respiratory substrate breaks down, but Krebs cycle could be considered as amphibolic because kisman bostu bhangi jay aru kisman important bostu synthesis hoy. When we talk about those 2 important intermediates that are formed during Krebs cycle, we are talking about succinyl coenzyme A and alpha ketoglutaric acid. Within that cycle, Krebs cycle kiman 8-step reaction. Tate ki produce hoy succinyl coenzyme A eta step or alpha keto producer. Why we have to study precisely about this too? Because they have very anabolic activities or tahat involved hoy ase, right. If you are talking about alpha keto, they play a role in nitrogen metabolism, and they are responsible for the formation of the first amino acid, that is glutamate or glutamic acid, right. As far as succinyl coenzyme A is concerned, we talk about the role of succinyl in the formation of chlorophyll molecule, right. So up till here we have done. Today what we will do is we will start the class with drawing that Krebs cycle, right. So first Krebs cycle to ami akilo. Ekhin loke clear hoise sobole? Krebs cycle or eta round of Krebs cycle or 3 ta NADH to produce kore. Eta jodi 3 ta hoy 3 ta 6 ta hobo niki? Kot 6 ta count korise? 6 ta ATP? 2 ta NADH opor to 6 tai hobo, kintu 3 ta NADH produce hoy, so 9 ta hobo. Car Krebs cycle boli kole no hobo no, you have to be precise. If you are talking about Krebs cycle, you have to talk about 2 rounds of TCA. Specifically jodi 1 round koa tetia hole eta hobo. Eta roundot keita produce kore 3 ta NADH2, eta FADH2, 1 GTP. You have that cycle? I mean, have you brought your NCERT book? Did you bring? Just see. In the meantime, we can draw the diagram, right? And do you remember this conversion, pyruvic acid into acetyl coenzyme A is helped by, it is helped by an enzyme complex to pyruvate dehydrogenase complex acile, and the reaction is called as what? Decarboxylation reaction. সেইকাৰণে pyruvic acid into acetyl coenzyme A কি produce কৰিব? Carbon dioxide produce কৰিব, and since we have that word called as dehydrogenase, so obviously reducing cofactor produce কৰিব. So এই reaction টোত কি হ'ব? এটা side carbon dioxide evolve হ'ব, কেইটা molecule 1 per pyruvic acid, আৰু এটা side কি হ'ব? 1 per pyruvic acid NADH to produce কৰিব. Is the point clear? Last classত আমি কৰিছো একিনি. Okay. While we are drawing the diagram for Krebs cycle, this diagram will give you some more intermediate steps that is not there in your NCERT book, but you can always study the cycle that is given in your NCERT book. ঠিক আছে, যেনেকৈ C's equanimity step to তাত নাই, ঠিক আছে. So either you can study this or you can study the NCERT book. NCERT bookত কি দিছে? মেইন কম্পাউণ্ড গিটাৰহে নামগিটা দি থৈ দিছে, মাজৰ গিটাৰ নাম দিয়া নাই, right? NCERT বুকত ডাইৰেক্ট সাইট্ৰেটৰ পৰা অক্সেলোসাক্সিনেট দিয়া আছে ন নেকি NCERT বুকখন চোৱা সাইট্ৰেটে কি ফৰ্ম কৰিছে সাইট্ৰেটে আলফা কিটো ফৰ্ম কৰিছে বাকিখিনি একো দিয়া নাই অক্সেলোসাক্সিনেট দিয়া নাই ওকে তো সাইট্রিক অ্যাসিড ডাইরেক্ট আলফা কিটো সাকসিনাইল আর সাকসিনেট ফিউমারেট ম্যালেট ফিউমারেট নাই ডাইরেক্ট সাকসিনেট টু ম্যালেট আর ম্যালেট টু অক্সেলো ওকে কিন্তু then I will suggest you study from this only. Kihor karone from MCQ point of view, from both's point of view, it's okay because if suppose a question is asked, describe the Krebs cycle and you draw this diagram, then it's okay. But from MCQ point of view, question to keneke ahe je NADH to kuntu step kihor pra kihor conversion out hoise. If you say succinate to fumarate ki produces FADH2, jodi fumarate to nopodha tetihale step to ami gomei napam, right? So you have to study this only. In this entire diagram, what is important? The stages where the reducing coenzyme are produced, right? Next is where carbon dioxide is produced, right? Next, if suppose carbon dioxide produced korise, so obviously decarboxylation hoise, right? Decarboxylation hoar loge loge carbon number to komi jabo. We have started from 6 carbon and ultimately we have obtained kiman 4 carbon. So, oh. Carboxylation mane carbon dioxide accept kora ulai joatu decarboxylation yate ulai goise no duita step and again we know that we have started it from 6 molecules or 6 carbon molecule ultimately oxaloacetic acid form korise hetu hol 4 carbon so obviously 2 carbon has been lost during the process in the form of carbon dioxide, right? So if this is one TCA cycle, please don't consider this portion if suppose specifically mentioned korise one TCA, right? Because this is nothing but the link reaction. We have just shown that acetyl coenzyme A enters or links up with Krebs cycle, right? This entire cycle is one TCA. So what is the product that is synthesized in one TCA? Ki produce kore? ATP produce kore na kore? Right? ATP keneke form kore substrate level mane ekdam simple language et ki buji pam the portion that we can see eto to dekhai paisu no GTP by ATP we can see it directly no this is substrate level. So question ahe what is the number of ATP synthesized by substrate level phosphorylation in Krebs cycle kiman hobo 1, right? Substrate level kole hodai 1 baki reducing cofactor bilake bhangi pelayu to ATP produce koribo, so that would, the question would be via ETS, right? Okay, so this is first. Next is talking about NADH2. How many molecules of NADH2 are produced? 3 molecules. How many FADH2? 1. So NADH2 kuntu step pra kuntu step ole hude or else the question I'll give you the question ebar ahise in which of the following step NADH2 is not produced. Saita option dise, etu option dise, etu option dise, etu option dise, etu option dise. In which step NADH2 is not produced? Because generally the question NADH2 produced to aha nai karon tinta ke to produce korise, right? So what is the point of interest is FADH2 kuntu step ot dehydrogenase enzyme to ase kintu FADH2 produce korise. Eneo what we need to remember whenever we see the word dehydrogenase obviously ki produce koribo reducing cofactor, dehydrogenase reducing cofactor, right? Next, we give the credit of evolution of carbon dioxide in Krebs cycle. What is the number of carbon dioxide evolved per TCA cycle and what is the number of carbon dioxide evolved in Krebs cycle? 1 TCA cycle or keta carbon dioxide ulaise, Krebs cycle or keta carbon dioxide ulaise? 1 TCA cycle or carbon dioxide keta ulaise? Duta. This one and there, right? Oh, sorry, this one and this one. Krebs cycle or keta aise? Krebs cycle will equal to 2 rounds, no? 4. So in the entire process, maximum carbon dioxide code ulaise Krebs cycle or 4 carbon dioxide evolved during Krebs cycle. So the credit goes to Krebs cycle as far as carbon dioxide evolution is concerned. Clear? ইয়াতো কিন্তু কাৰ্বন ডাই অক্সাইড ওলাইছে কিন্তু since it's just one molecule আমি বেসিকে যত ওলাইছে তাকে ক্ৰেডিটটো দিম so respiration ত কাৰ্বন ডাই অক্সাইড ফৰ্মেশন ত কোনে হেল্প কৰে it is a Krebs cycle that helps it clear চাৰিটা right and we have already talked about alpha keto and succinyl coenzyme right important kuntu anabolic pathway this one and that one right okay if this is over we'll write the question okay before wiping this out last class ot ami junkhini information likhisilu no hekini bostu swasun regarding the number of NADH2 and FADH2 produced 3 NADH2 1 FADH2 6 NADH2 2 FADH2 likhisu no points bilak hekini logot milai pelai swa whether it is understood or not only see the numbers okay reducing cofactor keita produce korise okol heikita points swa What is the number of NADH2 and FADH2 produced from one pyruvic acid? What would be the answer? 4. Kyo 4? Karan tinta yat, eta yat. So that is 4. What is the number of NADH2 or FADH2 produced by 2 pyruvic acid? Double hoi jabo, right? 8 hoi jabo titia. Or what is the number of NADH2 produced from Krebs cycle? 6 hobo. What is the number of NADH2 produced from one cycle or one TCA cycle or one Krebs cycle? It is 3 hobo. Jedi Krebs cycle diya double hobo, jedi 1 diye you have to be specific. Jedi 1 pyruvic diye then it will be 4 NADH2, jedi 2 pyruvic diye it will be 8 NADH2. Right? Ekita point clear hoise? I think I can drop the board. Ekita step mukhasto koribo lagibo de. Glycolysis or glycolysis of 2 NADH2. Pyruvic acid glycolysis harmonite produce hoy na shekhot produce hoy. Complete hoy joa pisat pyruvic acid form kora loke glycolysis is completed. So you don't go back. ঠিক আছে pyruvic acid or pura agwai gole link reaction aru Krebs cycle hobo. কিন্তু গ্লাইকোলাইসিসৰ যদি কোৱেশ্চন আহিছে it is from glucose to pyruvic acid only where you see only NADH2 how many 2 Ultimately, Krebs cycle What we are talking about here is Krebs cycle. If I use the word Krebs cycle, we understand we are talking about 2 rounds of Krebs cycle, right? There is single ke mention korne eta duta roundot keta poribo 6 4 2. How many carbon dioxide? 4. Ekto class no koriba, thik ase? Ha, jete ami glycolysis or class to korim, hedna tumi class no koriba. Thik ase? Tatsa gutei kon confused. Hoy ne? First year kabar kiba confusion ase glycolysis, is it clear? ATP clear, right? Kiman produce kore, 4 ta asile kio 2 ta consider koru, sob clear? Class no kori pelai to digdari hobo no. Okay, after the class we'll discuss again. Okay, let this class get over. Okay, so I think this is over. তোমার ভয়েস তো ক্র্যাক করেছে সাকসেনিক অ্যাসিডৰ আগৰ ৱৰ্ডটো অক্সালোসাকসেনিক অ্যাসিড ওকে Answer would be see just to have an idea say question to keneke hudise so it's important that you have to read the steps where the reducing cofactors are produced and the step where the GTP is produced and the step where the carbon dioxide are evolved right so ultimately gutei tu poribo laga hol right glycolysis or Krebs cycle we need to by heart over চাৰিটা হ'ব নেকি চাৰিটা হ'ব নেকি চোৱা ভালকৈ চোৱা কুৱেশ্যনটো অপশ্যনটো পিছত প্ৰথমে কুৱেশ্যনটো পঢ়া কি দিছে ভেৰি গুড হয় 12 কিয় কাৰণ এটা লিংক ৰিয়াকশ্যনৰ বাকী 3 + 2 + 1 হয় 3 NADH2 1 FADH2 1 GTP and +1 NADH2 from the link reaction so ultimately that brings a calculation up till 12 বুজি পাইছো নে নাই পাইছা Well, this is all about the first and the second step, that is Krebs cycle and glycolysis, right? Over. Yet is it confused? What? 4 + 12 whole, you can do the calculation. Come to pi sweet. Is it over? Online students? Okay. Is this understood? Okay. Okay. So since we have talked about the first and the second step of aerobic respiration, we have to talk about the third one, that is terminal oxidation, right? But before talking about terminal oxidation, we have to study something called as the electron shuttle. Now, I think all of us have a confusion as to what is the number of ATP molecule that is produced from one glucose. If one glucose molecule breaks down, then obviously ATP would be produced. Total number of ATP keta produced kore eta glucose. We have a confusion between 36 and 38. Right. So, we cannot be saying that it is exactly 36, neither we can be saying that it is exactly 38. So, 36 or 38 depends on what? Depends on which cell we are talking about. Kisuman cellot ki hoy glucose molecule to bhangi goi pelai 38 ta ATP produce kore aru kisuman cellot ki hoy 36 ta produce kore. Now, whether it is 36 or 38 depends on what? Depends on the one who is carrying the electron, right? So, we have something called as the electron shuttle. Cells bilakot electrons bilak carry kori NADH2 bhangi jabo no NADH2 bhangi pelai ki form korbo electron protons so electrons bilak carry kori etar pra etale dibo lagibo we'll be talking about that so tenekwa kisuman system ase jar karone either 36 or 38 ATP produce hoy from this section of electron shuttle basic details or poribo na lage obviously while writing a note we are going to give in little bit of details about it ami pura cycle to ba the pathway of the electron transport hebilako dekhai dim kintu examination point of view pra okol kataman sentence porim hegita sentence uke tick kori dim okol hegita poriba right from understanding point of view we are going to write something more. Right. So, electron shuttler, what is the importance of a electron shuttle? Suppose we are talking about this cell, right? Cell of ki hoisile glucose gets converted to pyruvic acid, right? Yate ki produce korisile NADH2 aru ki produce korisile ATP, right? Next is mitochondria, pyruvic acid, acetyl coenzyme, Krebs cycle, yate ki form korise clear? As far as this is concerned, NADH2, FADH2 and GTP is concerned, tahate bhangi pelai NADH2 aru FADH2 bhangi pelai ki produce koribo ATP produce koribo. Next is ATP production eta cellot kot hoy. Whereas ATP synthesis takes place in a cell, obviously in the powerhouse of a cell, so it's within the mitochondria. So, as far as these are concerned, no, tahator logot eku problem nai. Tahate code synthesize hoise mitochondria bhitorot bhangibo code, tate bhangibo ATP code produce koribo, tate produce koribo. Is the point understood? But we have a problem with the NADH2 that is produced in the cytoplasm. We know every reducing cofactor will break down and form ATP. So similarly, in case of glycolysis, the NADH2 that is produced will also have to break down and produce ATP. Now, while producing ATP, directly to yate bhangi pelai ATP produce koribo nware no, karon ATP production koribo lagile, we have already talked about chemiosmotic hypothesis. ATP produce koribo lagile duta requirement ase, one is the proton gradient and another is the enzyme. So this will break down, but yar pora ATP produce koribo lagile proper sequence ot ahibo lagibo aru kot ATP to produce hobo within the matrix of mitochondria. So this NADH2 that is produced in the cytoplasm during the process of glycolysis needs to be carried from the cytoplasm to the matrix of mitochondria so that it can form ATP inside. Is the point understood? Krebs cycle or jikita form korise tahator problem nai karon tahate jegate ase, right? Mitochondria bhitorte ase tate ATP form koribo. Problem to kot is problem to hoise juntu niki cytoplasm ot produce korise. Karon ami janu je ATP ketiao cytoplasm ot to produce no hobo no glycolysis or karone ei bostu to yale bhitor thumabo lagibo. So ei bostu to ki hobo bhangi jabo electrons aru protons, right? Electrons aru protons bilak eta proper pathway re jabo lagibo titia he ATP produce hobo. If you remember, you have, I think you have read about that ETS, right? You have that something like this, right? So the electrons and protons that are broken down here, no, itu bhangi pelai electron proton hole. Electron and proton kot ahibo lagibo, matrix of mitochondria ahibo lagibo, titia he tar pora ATP produce hobo, right? And this yale kune anibo, electron shuttle anibo. Some carrier molecules will bring the electrons from the cytoplasm to the matrix of mitochondria, and this is what is called as electron shuttle. Is the point understood? So, kiba doubts ase niki? Electron shuttle ekdom simple language at ki hoy carrier molecules ki koribo electrons bila carry koribo kiho karone koribo jate niki ATP form hoy kot ATP form hobo mitochondria bhitorot form koribo kor pora carry koribo cytoplasm or pora mitochondria bhitorle kunubai tak carry kori anibo he kunubator naam ki electron shuttle, right? So cellot bahut bilak basic compounds thakibo depending on who will take the electron. Accordingly, we have two pathways, right? Are accordingly either 36 or 38. Kunubai jodi loy, suppose we have A as a carrier and B as a carrier. Kun carrier we will discuss. Suppose in the cell we have two carriers. If the electrons and protons are carried by A, maybe then we'll have 38. If the electrons and protons are carried by B, then we'll have 36. Ekdom simply buzi lua. What is A and B we'll discuss later. Ekhilnoke buzi paisan nai. Clear? Last bench, clear? Online? Okay. So we'll write certain points about that. Electron shuttle portion to NCERT bookot nai di, kintu tothapi dutaman questions ebar ahise, ebar dubar ahise. Likhi loa first. Just read the line. Is it understood now? Sorry. Electron shuttle kyo lage? Just to carry the electrons and protons from the NADH2 from the cytoplasm to the inside of mitochondria. right? Our electron protons bilak jetia loke electron transport system that pathway through re najai tetia loke ATP produce no kore. ETS code ase. We know that the electrons and protons would be carried and they will enter the electron transport system. For those students who have already read the lesson, you should be aware of the fact that ETS buli eta pathway ase, carrier electrons and all those, right? Where is it located? It is located in the inner membrane of mitochondria. NADH2 code produces cytoplasm. He kole ahibo lagibo, inner membrane of mitochondria rakhibo lagibo, right? So kune help koribo, electron shuttle help koribo. Clear? It has to enter the mitochondrial matrix, but the membrane is impermeable to NADH2. Directly humai jabo nware, right? Jedi permeable hole heten cytoplasm pora NADH2 directly mitochondria bhitorot humai gol heten, but the membrane is impermeable, right? So you need certain help. So shuttle system keita duta shuttle system thake eta hol malate aspartate aru eta hol glycerol phosphate tamane electron to kune carry koribo malate malic acid carry koribo ne glycerol carry koribo jodi malic acid carry kore that would be malate aspartate aru jodi glycerol carry kore that would be glycerol phosphate. Details about this process or the cycle or the pathway we will draw in the class, but don't study from examination point of view iman nahe, right? But the points that we have to study from these we will be talking about it. As far as these lines are concerned, I think it's understood, right? So what is the importance of electron shuttle? What is the electron shuttle? Carrier system. What does it carry? Electrons. From whom? From NADH2. NADH2 from which place? In the cytoplasm. Keneke ahile? Glycolysis of RNA. Is it clear? Can I rub the board? Okay, last one likha upor to musi asu. Shuttle system is absent in case of prokaryotes. Kiho karone? Mitochondria na thake. No, shuttle system to kot thakibo. Inner membrane of mitochondria thakibo, right?
System carries the electron from
DH2?
or it carries the NADH2?
No, it carries the electrons from NADH2 to bhangi jabo. Right? Bhangi go play electrons produce koribo, electrons, protons, aru NAD le convert hoi jabo. Cytoplasm mote bhangibo. Glycerol, I mean glycolysis of carne jonto NADH2 produces cytoplasm what NAD. So ATP to janeke ADP le het cycle to soli thake no. Thik teneke reducing cofactor bila koru. NAD to NADH2, FAD to FADH2 cycle to soli thake. So NADH2 jodi cytoplasm produces he to convert ki hole hobo NAD, right? Ebar reduce hobo, ebar oxidized hobo, right? A cycle to code hoye cytoplasm on. Kintu hey electron khini carry kune koribo? Shuttle system e koribo. Right? Is it over? Okay. Eta next heading koriba mallet aspartate shuttle system. Mallet aspartate shuttle boli heading kora. NADH to ki hole convert hoise NAD code convert hoise first step ot chwa cytosol is cytoplasm, right? The NADH that was present in the cytoplasm gets broken down, electrons and protons by electron carriers oxaloacetic acid ki hole convert hobo malic acid transfer koribo, and this malic acid will help it to come inside the matrix, right? So they carry the electrons and put it in the matrix. Next is after malic acid gives out the electron to NAD, ki form koribo NADH to form koribo, and malic acid ultimately gets converted to oxaloacetic acid. Prothomote oxaloacetic acid kot asile cytoplasm ot, etia malic acid cytoplasm or kole ahile matrix hole, matrix ot malic acid e electron to DDR pisot ki hole convert korise oxaloacetic acid, kintu oxaloacetic acid is impermeable to the inner membrane, so he karone he directly ulai jabo nware, it has to get what with the help of ammonia, right? It has to get converted to aspartic acid, and only aspartic acid can go out from mitochondria, right? Outer membrane, inner membrane, matrix. Outer membrane of what? Mitochondria. Inner membrane of mitochondria aru matrix of mitochondria. Oxaloacetic acid, aspartic acid ole ki hoise ammonified, right? Tar pisot aspartic acid goes out. Ei chart khonor pora teneke questions aha nai de. If it's over, please say. Koriba. Rest of the students who have completed the drawing, is the portion up till now clear? Whatever we have discussed, clear hoise no? So depending on cell to cell, which type of a shuttle system is functional, it will vary. Kisuman cellot A2 shuttle system functional hoy, kisumanor glycerol phosphate shuttle system to functional hoy. Jedi A2 shuttle system functional hoy, then from one glucose the number of ATP that will get synthesized as 38. Kintu glycerol phosphate jedi kam kore tetiahale eta molecule opra kiman ulab 36. Eta kotha to hol kuntu cellot kuntu shuttle system e kam kore. If you are talking about heart cell, if you are talking about kidney cell, if you are talking about skin cells, right? Depending on cell to cell, the shuttle system is different, and accordingly the number of ATP synthesized would be different, right? Eto musu. So we'll write the points about this shuttle system. So shuttle system or kita porhiba, it is only these 3 points that you will study. ঠিক আছে? So kuntu efficient, we will be talking about 2 shuttle system, duitar bhitor efficient kuntu, june basically ATP produce kore hiye efficient hobo. So 38 june produce kore, it will be more efficient as compared to the other one, right? Next question ahe, heart cells, liver cells, maybe kidney cells, what is the number of ATP produced from one glucose? 38, right? If this is over, next to likha, glycerol phosphate. Code কিমানটা হ'ব হেতু ৰেসপিৰেশনৰ পিছত মই টেলি টু দিম ন এতিয়ালৈকে আমি কিমান পাইছো গ্লাইকোলাইসিছত পাইছো ক্ৰেব সাইকেলত পাইছো আৰু সৰি লিংক ৰিঅ্যাকশনত পাইছো ইফ ইউ আৰ টকিং অ্যাবাউট দ্য নাম্বাৰ অফ ATP সিনথেসাইজ কোনটো স্টেপত ম্যাক্সিমাম হয় ইট ইজ অলৱেজ ইন দ্য টার্মিনাল অক্সিডেশন and again from the this one if suppose if you have read about ETS এটা NADH টোৱে তিনটা কিয় প্ৰডিউস কৰে আৰু এটা FADH টোৱে দুটা কিয় প্ৰডিউস কৰে you should have the answer why আমি তো নরমালি কৈ আছো ন এটা NADH টোৱে তিনটা প্ৰডিউস কৰে এটা FADH টোৱে দুটা প্ৰডিউস কৰে but there should be a reason behind that also so what would be the reason for those students who have read this chapter before something that talks about the ETS কি হয় in a mitochondrial membrane or কিছুমান complex আছে depending on the electron moves to which complex if it moves from the first complex onwards তেতিয়াহলে তিনটা ATP produce কৰিব যদি first complex ত যাব নোৱাৰে second complex ৰ পৰা তাৰ journey টো আৰম্ভ কৰে তেতিয়াহলে দুটা ATP produce কৰিব NADH2 can enter from the first and go to the all complex FADH2 cannot enter the first complex এই হদাই second complex ৰ পৰা journey আৰম্ভ কৰে হেকাৰণে number of ATP produces less আমি ডিসকাস কৰিম এতিয়া ঠিক আছে এটো হল ওকে গ্লিসারল ফসফেট লিখা সো এই তিনটা লাইনহে পঢ়িবা দেই বাকি বিলাক না লাগে In between inner membrane and outer membrane, there is a space. Space to kami ki kom? Perimitochondrial space. So this is the perimitochondrial space. What happened? এটা দুইটা shuttle systems যদি compare করা, what is the similarity? The similarity is both the shuttle system are carrying the electrons from the cytosol to the mitochondria. Similarity. What is the difference? Difference is, আৰু এটা difference কি যে এটা shuttle system electron to নি পেলাই from the cytoplasm electron to নি পেলাই ultimately কি form কৰিলে NADH2 produce কৰিলে. But if you are looking into this one, NADH2 পৰা electron to নি পেলাই কি form কৰিছে FADH2 ক'ত form কৰিছে ultimately এটো তো ইয়ালে আহি যাব ন FADH2 তোমাৰ matrixৰ পৰা ইয়াত উলাই গৈ পেলাই ইয়াতে electron protons ভাঙি পেলাই ETS ত যাব ETS ক'ত আছিলে inner membrane তে আছিলে. We'll discuss later. As of now, is this point clear? Similarity is in both the cases the electrons from the cytosol from NADH2 is carried to where? Carried to the mitochondria. Next, in the first one malate aspartate that কি form কৰিছিলে electron to নি পেলাই ultimately NADH2 produce কৰিছে ইয়াতে কিন্তু electron to নি পেলাই কি produce কৰিছে FADH2 আৰু যেতিয়া FADH2 produce হয় তেতিয়াহলে number of ATP synthesized is less যেতিয়া NADH2 produce হয় number of ATP synthesis is more. So this shuttle system will give you 36 ATP, that shuttle system gave us 38 ATP. Clear? Oh, I did not write that cytosol, no? I think this part we can rub. Nahi hua? Ako likhi দিব লাগিব নি? Over? হলে নাই তোমাৰ? Sorry. So from glucose, one glucose, either 36 or 38 ATP are synthesized. Okay, is this over? Next likha terminal oxidation, the third step. You laugh before asking a question. Ek second. Tumar karne mo spelling bhul ho gol. Okay, now say. Ki kosa? Basic as the ATP cell, ATP form or brain cell or no brain to phati jabo. Right? Mane hey like a battery paisa niki mane basic charge thaka battery kom thaka battery, right? So the first energy to obviously protect to cell or lagibo kintu basic energy also it's not good na. Basic energy hole cell to moriye jabo dokhon iman basic. That is the reason why in the process of respiration eta glucose bhangile potokke 36 ta ulai nahe stepwise, so that it does not harm the cell, right? Gradual. Okay. Tumi to mask khon gusoa ma khuna nai. Okay. Brain. No, plant or thik teneke nohoy. We are talking about this from animal cell point of view. Karon glycolysis and Krebs cycle tato to hoy no. So combination say cellular respiration process. So we are talking from the animal point of view. Right? Okay. Okay. Now terminal oxidation. Terminal oxidation. First, what we'll do is we'll understand the process and then we'll start writing. Okay. So up till now, what we have said is like in the process of respiration, what happens is glucose gets broken down. Glucose bhangi pelai কি ফর্ম কৰিব পাইৰুভিক এসিড তাৰপিছত কি ফর্ম কৰিব ক্ৰেবছ সাইকেলৰ পিছত থাৰ্ড ষ্টেপটো কি হ'ব টাৰ্মিনেল অক্সিডেশ্যন গ্লাইকোলাইসিছত কি প্ৰডিউস কৰিব অনলি এটিপি ক্ৰেবছ সাইকেলত কি প্ৰডিউস কৰিব এটিপি এণ্ড কাৰ্বন ডাইঅক্সাইড right as far as the third product is concerned jar karone ami complete oxidation boli kobo parim that is we are talking about the evolution or production of water the credit goes to terminal oxidation so ultimately terminal oxidation ot ki hobo terminal oxidation ot water produce hobo right e jetia terminal oxidation reactions bilak solibo ultimate product hisabe ki produce hobo water kune produce koribo the protons plus the oxygen together will form H2O is the concept clear hoboi lagibo no water form korile H2O to H aru O lagibo so oxygen was already present right code present will discuss tar logot proton part to log lagi pela ultimately ki form koribo water form koribo next terminal oxidation is also a step of respiration so if it is a step of respiration then ATP needs to get synthesized so terminal oxidation is a process that has two steps first the formation of water and second the formation of ATP right the first step is something called as a electron transport system and the second step is called as oxidative phosphorylation terminal oxidation duta step ot hoy eta ETS second okay oxidative phosphorylation ETS or karone ki produce hobo water terminal oxidative phosphorylation or karone ki produce hobo ATP right so terminal oxidation or keita step hobo eta hobo ETS aru eta hobo oxidative right so ETS ot ki produce hobo water Oxidative phosphorylation or key produce hobo. So A2 section of how is water produced, how is ATP produced, we have to talk about. Clear? Okay. Now terminal oxidation kot hobo. Mitochondria te hobo. Mitochondria hobo noy. Mitochondria te hobo. Right? So mitochondria hobo lagile. Etia ami ki dekhilu se inside the cell, glycolysis hoisile, then we have the mitochondria, what we have the reducing cofactor bilak ase aru energy ase. Energy has been used up, right? Jonto GTP form korisile no, GTP to use kori dibo cell to tate to roi na jai. Respiration or karne jiman bilak energy produce hoise cytoplasm ote hok ba tumar mitochondria matrix of bhitorte hok, what will happen to that ATP produced? Obviously it will be used up by the cell for various metabolic activities. So ATP to teneke store hoi accumulate okol hoi na jai no, tak to use koribo. So talking about the GTP that is produced in the Krebs cycle, it will get used. What is left behind? The reducing cofactor. Matrix of key form korisile. Eto jedi matrix of mitochondria hoy, yate key form korisile. Krebs cycle. Krebs cycle okane ki form korisile. NADH2, FADH2, GTP. GTP is energy, it is used hoi jabo. So ultimately matrix of bhitorot ki ase etia? NADH2 or FADH2. Matrix of bhitorot reducing cofactor ase. Jedi matrix of bhitorot reducing cofactor thake, then we could have used a statement saying mitochondria is the house of the reducing cofactors. Ami kau niki teneke? Ami ki kau? Mitochondria is the house of ATP. It is not a house of NADH2 or FADH. Ihan ta Bangladeshi nisna hoy na hoy. Lagibo jano ta? Are they wanted inside? No. What they want? Only ATP. So this too needs to get broken down and form ATP. Is the point clear? So terminal oxidation or karone junto ETS form kore. ETS mane the electron transport system. Electrons will move from one place to another, and that is what is called as electron transport. The first point is, from where will they get the electron? Electron to move koribo lagile electron to kobar pra ahibo lagibo. Electron to kor pra ahibo? NADH2 or FADH2 pra. Kihor karone matrix or bhitorot junto NADH2 FADH2 produce hosile. That NADH2 and FADH2 cannot remain in that form because it is not their house. So what will happen? They will get broken down. If NADH2 gets broken down, key form koribo electrons, protons, NAD. If FADH2 gets broken down, key form koribo electrons, protons, FAD. So within the matrix, if suppose we are talking about NADH2, key form korile electron, proton, NAD, right? Aro FADH2, electron, proton, FAD, right? So ultimately what we can say, NADH2 and FADH2 gets broken down inside the matrix of mitochondria. Clear? Next, jetia eta bostu ebilakole bhangi jabo, electrons and protons needs to be separated, because if they stay like this, akko to NADH2 form koribo. Remember, this is a concept we have talked during photolysis of water also. Water molecule jete electrons, protons ole bhangi jai, the electrons and protons needs to get separated. They cannot be captured like that. If they are captured like this, they will combine together and form the product again, right? So for that, what they have to do is they have to remove this electrons and protons. So mitochondria bhitorot electrons and protons bilak remove koribo lagibo. Remove korile kole jabo. So if you are talking about mitochondria as outer membrane, inner membrane and matrix. NADH2 code form korise matrix or FADH2 code form korise matrix or tahate bhangi pelai ki form koribo clear, right? So electrons and protons need to be thrown out of the matrix. Tahatok matrix apra pothai die. Now when they are not thrown out from the matrix, tahat kole ahibo yale ahi jabo na jai, matrix apra ulai dibo, right? So electrons and protons will be thrown out from the matrix, and then they will come to the inner mitochondrial membrane. Inner mitochondrial membrane or ketaman complex ase, ghor ketaman ase. Keita ghor ase? 5ta ghor ase. ঠিক আছে, we'll understand the concept and then we'll write the notes. Notes লিখাটো ইমান বেছি ইম্পৰ্টেণ্ট নহয়, বস্তুটো বুজি পাব লাগিব। তো এতিয়ালৈকে আমি কি কৈছোঁ যে মেট্ৰিক্সত যোনটো NADH2 আৰু FADH2 প্ৰডিউস হৈছে তাহাতক কি কৰিব লাগিব ৰিমুভ কৰিব লাগিব ৰিমুভ কৰিলে তাহাতে কোলে আহিব right so ইয়াৰ পৰাও কি হ'ব ইলেক্ট্ৰনছ প্ৰোটনছ আউট হৈ যাব ইয়াৰ পৰাও কি হ'ব ইলেক্ট্ৰনছ প্ৰোটনছ আউট হৈ যাব right এতিয়া ইলেক্ট্ৰনছ এণ্ড প্ৰোটনছবোৰ ইয়াৰ পৰা আউট হৈ গ'লে ইনাৰ মাইটোকন্ড্ৰিয়াল মেমব্ৰেনত কিছুমান ইলেক্ট্ৰন একসেপ্টৰ থাকিব লাগিব তেতিয়াহে ইলেক্ট্ৰনছবিলাক ল'ব নহলে কিয় ল'ব তোমালোকৰ ঘৰত নাজানা মানুহ এটা আহি যাব তুমি কি দৰজাখন খুলি দিবা নেকি obviously right you have to know the person and then only he'll get inside right the first point is inner mitochondrial membrane ot ki thake ketaman complex thake how many complex 1 2 3 4 5 right complex mane ki protein molecules right a complex bilak jodi ami ghor hisabe consider koru yate to ghorot to manu thakibo no so ghor bilakot ki thake components থাকে inner mitochondrial membrane ot ki thake complex bilakor bhitorot ki thake components thake what are the components are electron carrier right so yate kisuman enzymes thakibo various enzymes will be present in the complex and accordingly what they will do is they will take the electron and transport it to the next cell এটো যদি গুটেই বিল্ডিং হয় তেতিয়ালে এটা রুম দুটা রুম তিনটা রুম চাৰিটা রুম পাঁচটা রুম রাইট এই আউট অফ দিস ইয়াতে সবতে কিবা কিবি কম্পোনেন্টস আছে কম্পোনেন্টস বিলাকে কি কৰিব ইলেক্ট্ৰন টু ট্ৰান্সফাৰ কৰাত হেল্প কৰিব accept and transfer right now if the electrons and protons are thrown out no it is not like this that they will come out single eta electron ahile eta proton ahile teneke nohoy they always come out in a team and the team comprise of 2 electrons and 2 protons matrix or pora jete electrons proton push out kori dibo tetia tahate ki hisabe ahibo team hisabe ahibo the team consists of ki 2 electrons and 2 protons will always come out together from the matrix to the inner mitochondrial space clear so ami etu buji palu keneke ulai teamot ulai next first we'll be talking about NADH2 NADH2 jetia electrons and protons ulai dile ei complex tule ahile first complex ot ki ase they have a enzyme or they have a enzyme called as NADH2 dehydrogenase right iman complicated na lage yate ki hobo junbilak electrons ahibo electrons bilakok tahate sini pai he karone tahatok khumabole dibo যদি ইলেক্ট্ৰনছ NADH2 ইলেক্ট্ৰনছ কিটা চিনি না পায় তেতিয়াহলে ইহতে কেতিয়াও খুমাবল দিয়ে the simple way of understanding is you will allow a person whom you know to enter into your house and not a stranger so NADH2 যোনবিলাক ইলেক্ট্ৰনছ and protons আছে ইয়ালে আহিব ইয়াতে কি আছে ইলেক্ট্ৰন acceptors and carriers আছে so ইয়াতে যোনটো ইলেক্ট্ৰন acceptor আছে তাহাতে কি কৰিব এই ইলেক্ট্ৰন proton খিনি লৈ লব তাৰ পিছত electrons khini next complex ole pathai dibo protons to push out matrix or pra ulaute keneke ulaise teamot electrons and protons together where they have reached inner mitochondrial membrane ot ki ase electron acceptor ne proton acceptor electron acceptor so will they accept the protons no but while they were coming out they came out in a team right so ahute to teamot ahise accept kune koribo okol electrons to protons khinir ki hobo push out kole push out koribo is the concept clear
tite matrix electron and proton asbe tite to mane eklog hoda onali inner membrane dalito ekelogi anilo tite to eklog hobo paribo
নোৱাৰে তাহাঁতৰ লগত কাৰণ NAD টো নাই ন and electron proton NAD complete হলেহে NADH produce কৰিব তেবেতে NAD বাহিৰৰ ভিতৰতে থৈ আহিলা অকল ইলেক্ট্ৰন proton খিনিহে ক্যাৰি কৰিছা right okay understood ha
proton acceptor to
নাই
so proton to keneke আহিব pare
নহয় আহুতে ইয়ালে তো খুমাবই খুমাই পেলাই দুটা মানুহ আহিছে তোমালোকৰ ঘৰলে দুটা মানুহ কলিং বেল বজাইছে তুমি ভিতৰত কাক সোমাবা যাক চিনি পোৱা বাহিৰটো কি কৰিবা আউট এক্স্যাক্টলি দ্য সেম কনসেপ্ট ৰাইট আহোঁতে দুটা আহিছে কিন্তু তুমি ৰুমৰ ভিতৰলৈ কোন যাব যোনে চিনি পায় স্ট্ৰেঞ্জাৰ এটাক তুমি ৰুমৰ ভিতৰত নোসোমোৱা তাক পঠাই দিবা সো এক্স্যাক্টলি তাহাঁতে কি কৰে এইটো কমপ্লেক্সৰ পৰা সেকেন্ড কমপ্লেক্সলৈ ইলেক্ট্ৰনছখিনি পঠাই দিয়ে হা প্ৰত্যেকটো ৰুমত যাব পাৰে ৰাইট আৰু প্ৰটনছবিলাক দে উইল বি পুশড আউট আপ টিল হিয়াৰ ইজ ইট ক্লিয়াৰ Now this is not a very scientific explanation, okay? Scientific explanation hole to we have to go by the sentences that are written in the book, right? This is a way to explain and understand the process, what happens exactly, right? So electrons and protons are pushed out, electrons moves through the complex and the components, the protons are pushed out. Protons will push out kore pelai kot accumulate kore perimitochondrial space. Clear? Now from if you are talking about this electron will kot move koribo from one complex to the other complex. Inner mitochondrial membrane or keta complex ase pasta. Out of the 5 complex, it is only 4 complex through which the electron can move. 5th complex or electron ahibo nware. Clear? So electrons first or ahile, first or second or ahile, second or third ahile, third or fourth ahile, fourth or fifth ole ahibo niki? Nahe. Eta room locked. Right? Okay. Electrons not allowed. Kitchen ko aji ko. Actually kitchen ole ATP produce hobo. Anyway, chalo understood now? Now while they are doing this, what happens is one NADH2 will push out 3 pairs of protons. Eta movement ot jetia complex hole ahi thake tetia tahate proton movement kebar koribo 3 bar. Eta NADH2 bhangi pelai ki produce korile electrons and protons. Electrons and protons bilak timot ahi pelai kole ahile inner mitochondrial membrane. When they move from one complex to the other complex, what they do is they will push out the protons. How many times they will push out? They will push out for 3 times. That is the reason why NADH2 produces 3 ATP. ATP production are responsible, protons are responsible. We'll talk about it later. As of now, understood? Okay. Come to FADH2. FADH2 will also break down and produce electrons and protons and FAD. Electrons and protons will also come out in a team. Right? Now this team cannot enter in the first complex. They don't know. Tahate kot enter koribo, hodai they will start their journey from the second complex because tahatok sinipua enzyme to FADH2 dehydrogenase kot ase yat. NADH2 dehydrogenase kot ase yat. If that enzyme is present, it will recognize the electrons of NADH2 only. If FADH2 is present or FADH2 dehydrogenase is present, then it will take the electrons from FADH2. As of now, what we have understood, electrons of NADH2 can come from first complex to the fourth complex. Electrons and protons from FADH2 can start their journey only from the second complex to the fourth complex. 4 number loke U ahibo NADH2 eta ghor basically ahile nai ahise so tar ATP production besi, right? Next point, NADH2 when this team came in, how many times they have pushed out the protons? 3 times. If FADH2 in case of them, if the team comes out, then they will push out protons only for 2 times. Right? Eta NADH2 kiman pairs of protons? 3 pairs of protons. Eta FADH2 kiman pairs of protons push korile? 2 pairs of protons. That is the reason why eta NADH2 pra 3 ATP, eta FADH2 pra 2 ATP. Is the point clear? Everything depends on the ETS. Clear? Okay. So this is something that's understood. What we have to read is the name of the complex and what are the components. Question ahe, first complex or component kun hoy? Second complex or component kun thake, third complex or kun thake. Yate ki thakibo? Electron carriers bilak thakibo, no? And whenever we talk about electron carriers, one common type of a carrier that we have already talked about is, if you remember, we have talked about cytochrome. Right? Cytochrome koisilu, cytochrome or kam ki electron transfer kore na eta jekar pran. So enukwa eta complex ot cytochrome complex o thakibo. Cytochrome molecule thakibo. Cytochrome bilake ki koribo? Electron bilak etar pra etale nibole help koribo. When we talk about cytochrome, that is said to be a complex and not a single cytochrome. So we have a collection of cytochromes, cytochrome C, C1, A1, A3 and A. right? So ultimately yate C thakibo, C1 thakibo, A thakibo, we'll draw that later. Last thought, in the fourth complex ki thakibo, cytochrome A3 thakibo, right? So electron bilak components ole ahi ultimately electrons code jabo cytochrome A3. Cytochrome A3 is very important karon question to ahe last thought electron to kole ahe, last thought electron to cytochrome A3 le ahe. Second is cytochrome A3 e electron to jete accept koribo, they will release it also, they will not keep it along with them. Jedi thoi die tahato negative charge ho jabo no, so tahate ki korbo accept kori release kori dibo. So what they will do, they will push the electrons to the matrix. Electrons kor pora ulaisile matrix or prai ghuri meli phuri pela ako bhitorle humale. Clear? A electron to kunbai yat accept koribo lagibo oxygen, ultimately ki form koribo is the point understood? Nware tar pora kyo lobo nware karon jedi NADU ase no, NADU ase electron protonu ase, jedi gutei khini ekelege thake NADH2 form kori dibo, kintu etu system hor karone ki hol soson protons bilakok separate kori dile no, okol electrons kini ha move korise, that is the reason, right? So up till now is it clear? Terminal oxidation or ki hoy duta kam hoy, water produce hoy, ATP produce hoy. Water production or karone kun responsible? Electrons responsible. Whose electrons? NADH2 FADH2 electrons. Kor pora ahe, matrix or pora ahe, kole ahe, inner membrane ole. Inner membrane or ki ase? Complex and components. What they do? Take the electron, carry and transfer it to the next complex. Ultimately, at last, the electrons enters or is taken in by cytochrome A3. Cytochrome A3, where is it present? Inner mitochondrial membrane. What does it do? It transfers the electron from the inner mitochondrial membrane and push it to the matrix. Once the electron enters the matrix, kunubai tak accept koribo lagibo. Kune accept koribo? Oxygen. And oxygen will combine with that electron and ultimately what they will form is water. Right? Next, we have talked about the journey of the electrons, but we know electrons bilak to okole aha nai, tahate to protons or logot ahise. Ahute okol electrons ahise ne team ahise. Team ahise, ami iman khini ki porilu, transfer of electrons. What will happen to the protons? The protons also needs to do something, no? Yate to accumulate hoi thakibo nware, right? Now, we have seen accumulation of protons in the perimitochondrial space. Accumulation of protons leads to the formation of dash proton motive force. Proton gradient form koribo nokore. Yate all of a sudden proton basic ke hoi gol na nai. Kyo besi hoi gol? Karon NADH2 pora tinibar push out korile, FADH2 pora dubar push out korile. As a result, this area had more number of protons. Eta jagat less ase, eta jagat more ase. This is what is called as ki proton gradient. Proton gradient leads to what? Proton motive force. And when there is a force, they will push out. So these protons will again be pushed out. Yar pora kole jabo. These protons will enter the fifth complex. As I said, it has 5 complexes. Out of the 5 complex, 4 complexes are associated with the movement of electron, and the 5th complex is associated with the movement of proton. Right? 5th complex at electron jabo nware. It is only the proton that they will move. Or you have to refer it to chemiosmotic hypothesis again. So if there is a proton motive force, the protons will be pushed out. Right? Ami photosynthesis porisilu no e bostu tu je yata jetia protons pur besi hobo teta tara push out koribo. Kuntu through push out kore? FOF1. So this complex is equivalent to FOF1 particle. Right? So protons bilak ki hobo? Push koribo. So this is FO and like this, right? So ultimately protons bilak ahibo, F1 partot ahibo, F1 partot ki form koribo? ATPase, protons bilak kole push koribo. Right? So A2 lole ultimately ki form korile. Right? Aru plus ATPase ase jihetu yate ki form koribo. Eta duta question. ATP code form korise, matrix of mitochondria. Water code form korise, matrix of mitochondria itself. Is the point understood? So this is terminal oxidation. As far as the writing portion is concerned, we'll write it in the next class. Etialoke ami ki buzilu? Terminal oxidation is a third step of respiration that will make the process complete, right? Credit goes to what? Credit goes to production of ATP and production of water. Water, who is responsible? The electrons. ATP, who is responsible? The protons. Electrons, protons, what is the source? The reducing cofactor. Kot asile? Matrix of mitochondria asile, right? When NADH2 and FADH2 gets broken down, the electrons and protons are pushed out from the matrix to the inner mitochondrial membrane, and when they are coming out, they always come out in a team that consists of 2 electrons and 2 protons together. The 2 protons will move from complex 1 to complex 4 and ultimately would be accepted by oxygen in the matrix of mitochondria, right? As far as the protons are concerned, protons would be pushed out in the journey. Right, NADH2 pushes out 3 times, 3 pairs of proton push kore. FADH2 starts the journey from the second complex, ultimately it goes to the 4th complex and gives the electron to oxygen. Aru, how many times they will push out the protons? Is 2 times, right? And so that will lead to the formation of 2 ATP per FADH2, 3 ATP per NADH2, right? As far as the protons are concerned, because of the pushing out nature, no, the protons will get accumulated, they will form something called as a proton gradient. Proton gradient will lead to a formation of a force that is called as proton motive force. Because of the force, the protons will be pushed out to enter inside the matrix. When they will go again back to the matrix, they cannot go back through these complexes. First, second, third, fourth. Egita car complex, electron complex, proton jabo noare. So proton kunture jabo. 5th complex. So 5th complex is also called as the pathway of protons, right? So yar pra goi pla yat goi ultimately water form koribo. In the meantime, tahade ki form koribo? ATP form koribo. Clear? I think that's all for today. Thank you. As far as the writing is concerned, we'll write. This is something that's very important. Ekhine to questions ahe plus complex bilakor naam aru complex or components bilakok dibo, right? NCERT book ekhon diagram ase no, that pathway. Hey diagram to sabasun. Diagram to first step ot ki likha ase sosun ekdom first circle to. This diagram, yeah. First ot ki likha ase FMN ase nai? Ase FMN e electron to kak dise? FES, right? FES or pra UQ, UQH2. So that is the pathway that you have to remember. So NADH2 electron to kune accept koribo FMN. FMN kot thake first complex. That is the reason why the electrons move to the first complex. If you are talking about FADH2, FADH2 kune accept korise? UQ, UQ ase. So UQ is present in the second complex, so the electrons move to the second complex. Right? This part we'll discuss it in the next class. All right. Thank you.